avoid being rich. Alright? Okay, and 3, 2, 1, go. Who captured any? Again, we'll try one last time. 3, 2, 1, go. Alright, so we're gonna start our afternoon. The madam here. And I'm going to welcome Professor Anne for a talk, and hopefully we're all rejuvenated back from lunch. Karibu.
So, and asante sana kwa chakula for the lunch.
Thank you for the
perfect.
For the food. Yes.
So I would just say I would like to really thank the organisers for this fantastic opportunity to be here on your inaugural conference. So I'm now hoping that the amazing people who do the AV will bring up my title. But while they're doing that, in order to keep on time, and I know Prof is doing an amazing job to keep us on time. Right, there it is. So, my very good friend and colleague, Professor Mashidi, gave me the talk to talk about collaborative physiology research within and beyond Africa, which I thought was extremely challenging because, I haven't really thought about this category, but then I thought about it for a little while, and so I found a way that I think I'll tell you about the science that I've learned, and also, hopefully, actually come back to the collaborative physiology research and actually catch some of the ideas that were presented earlier. And really, I want to tag along on the 2 talks that were just started. And Dr. Abdulatif, who really said, is physiology important? And I want to agree with him and say that it is indeed really important. And also, Professor Essel, Professor Judith Zira, who I share the lab with, and who was actually up here before because she has been one of the members of the Nobel Committee. She's still on the Nobel Assembly, but she's not on the Nobel Committees. And so, I want to take you a challenge that was really posted by Professor Essel, when he said, you can study something boring, like exercise. Now that's what I studied, and I'm trying now to take this challenge and prove to you that it's not boring at all. Which is makes and a challenge, and it's now just after lunch, it goes practical. So let's see if we can actually get this to work. So the example of an exercise bout, and I'm only going to talk to you, well not only, I will talk to you a little bit about it, but about skeletal muscle. So why skeletal muscle? And this is just now a reminder, sort of a bit of physiology 101. And that is that now, as we all sit here and we have eaten and it's postprandial, where is our glucose going to be disposed? Really, we have an insulin release, and here is a very old study. Now, some of the people in the back of the room, you weren't even born when this study was done, but here it's where glucose goes after a meal. And here at the bottom is the brain, so that's always receiving a lot of glucose. This big yellow part is skeletal muscles, that's what I'm going to talk about. And the reason is not just that it's the biggest glucose disposal organ, but it's also, in fact, here on the right, we can see that if you have type 2 diabetes, that's when the glucose is not going anymore. So there's a significant impairment in the ability of skeletal muscle to dispose of glucose in people with type 2 diabetes. And just to then remind you, it's also a very large organ. Here we have sort of just one person. Most of us, we have between 40 and 60% of our body is skeletal muscle. So it's a significantly large organ. Now, why am I going to talk to you about exercise? Well, I'll go over here and see what I'm saying. So why exercise? Well, again, we're back to a paper that's published in 1988. And what you see here is a glucose challenge curve. So basically what you do when you want to prove that someone has diabetes, so basically see how well does your body handle glucose. You give a glucose challenge in the fasting state and you follow glucose excursion over 2 hours. And the curves here, the one at the top, is in 7 men with mild type 2 diabetes at the start of the study. And the study was a 7-day exercise intervention. And after 7 days, the investigators did the same again. And here is a glucose challenge curve. after 1 week of exercise. And you can see what the intervention was. It was 6 days of supervised exercise, 1 unsupervised, pretty intense exercise. But despite the fact that we now have semaglutide, SGLT2 inhibitors, all these things, there is really no medical treatment today that has this kind of efficacy. So why don't we use it more? This is a question more for students of psychology and human behavior, but clearly we know this works. It doesn't work for everybody. This we will get back a little bit to the idea of precision medicine. In these ones, it worked in 5 out of 7, but you still got the stats. And in 1988, you could actually publish papers with just 7 people. It's not really possible anymore. And the reason why it only worked and why it didn't work in 2 of them was that they actually already had their beta cell failure, so they couldn't improve their insulin sensitivity. Because that is what happens. In response to an exercise bout, what happens in your muscle? Well, acutely now, if we had exercised a bit more when Thomas was getting us to do hand lifting, what happens in the exercising muscle is that you have an acute uptake of glucose, even in the absence of insulin. You have acute muscle glucose uptake. But you have a lot of other things as well. You have a disruption of homeostasis. And we have the increased energy demand, and we have enhanced insulin sensitivity. And even one single exercise bout will significantly improve your whole body insulin sensitivity, or actually it's only in that muscle that exercise really, for several hours afterwards. And then you have the chronic effects that I'm not going to talk about. We'll talk about the acute effects, but chronic effects are somewhat the sum of many, many different small acute exercise bouts. So if we want to look ahead, so we already knew this in 1988 that exercise is a highly efficient way of improving your insulin sensitivity, of improving your type 2 diabetes. So what are the challenges? Now, in contrast to Dr. Abdullah, I'm not a purebred physiologist. I just started out as a molecular biologist, and I've spent the past 20 years trying to become a physiologist. But if we just put the question that I've been trying to study for a long time is, we have muscle here on the left, and we have the training muscle, which has more increased insulin sensitivity, strength, endurance. It's metabolically flexible, means it can use lipids even when you are performing exercise, and has an increased number of mitochondria. And the only input is training. The input is training. So what is training doing? So clearly it has to do something to remodel the exercise metabolically and functionally. We all know it remodels functionally here, because if we want to run a marathon, we all train, because we have the functional remodeling, we have a metabolic remodeling as well. How does it happen? Well, it could be transcription factors, could be different metabolites, histone modifications, DNA manipulation, non-coding RNA, and that's basically what I want to tell you about. How are we trying to understand that? But first I want to give a little vignette from one of my colleagues, Nico Pilon, and just show you the power of many small studies and how they can work together. And what Nico did, he's also works with exercise physiology. He mined the data, the public data that's available on human exercise intervention trials, and basically downloaded all the data that's there. It's free, it's out for all of us to use. But what Nico has done, he's made a database with all these various studies. And so it's also available to anyone who wants to use it. So write down metamex.eu, because you can use it for your own favorite gene. But what Nico did was, he basically looked at all these acute aerobic studies, training aerobics, so you do many exercise bouts, acute resistance, or training resistance, so more like hypertrophy, or inactivity studies. And he put them all together, and he basically cleaned the data up so that you can compare it. So actually, if you go online on the metamex.eu and you put in your favorite gene, you can then select, I want to only have the acute exercise studies, or only the acute resistance studies, and it will tell you if your gene is modulated. And just to tell you a little bit, if you just do the global whole picture analysis, here's the principal component analysis, and what you can see here, the orange is the acute resistance, and the yellow is the acute aerobic. And you can see that they segregate away from the training effects, which are more over here. So it's more the duration of the exercise than what kind of exercise you did. But anyway, it's just a little plug to say. Now here's just to remind you of the fact that when you do exercise and you move your muscle, clearly a lot of different systems are activated. And here's just from a recent review written by my colleague Janine Zira of all the things that we already know are activated. But the question is, how is it cognitively regulated? So now I want to take you through an ongoing study that's going on in our laboratory at the moment. Some of it's a little bit published, but really it's all based on one sort of study paradigm. And I've just divided it into three. It's muscle exercise changes in mRNAs, in skeletal muscle adipose tissue crosstalk, and a little bit about metabolomics at the end. So this is the setup that we use for all these three studies or vignettes. And we've recruited people who either have normal glucose tolerance or have type 2 diabetes. And they come into the lab in the fasting state. And we take a biopsy. And then they get on a bike and they exercise for 30 minutes. And we've actually screened them a little bit earlier so that we know exactly what's 85% of their maximum heart rate. So they do that for half an hour. They get off the bike and we take biopsies again. And the samples we take are muscle biopsies, fat biopsies, but we do that in the same incision. So on the way into the muscle, we actually take the subcutaneous fat, and then we also take blood. And here is if you have type 2 diabetes, and here you have type 2 diabetes 3 hours later. Now it's a bit difficult to appreciate from here, but what was really striking is that the number of genes that changed in somebody, and it's only men here and the study, the number of genes that changed in men in response to exercise was about 500. It's over there somewhere, 537. The number of genes that changed in someone with type 2 diabetes was 4,500. So a whole different order of magnitude. Which, and this was at the 3-hour time point that we saw this. But it's really surprising, thinking, at some point, what's the exercise doing in a person with type 2 diabetes that's so very different? And this was published last year, so you can, if you're really interested, you can read the paper, all the fancy analysis. But basically, what we see, or what we think we see, is that in the person with type 2 diabetes, it's not evident at the resting stage in muscle, but after the exercise, there's a huge call for inflammatory cells. They seem to be flocking into the muscle and then changing the whole transcriptomic landscape. And we don't know whether this is a good thing or a bad thing. Or quite what the implication is, but clearly it's quite different. And as a matter of fact, we then, Nico, looked using immunocytochemistry at CD11 plus cells here. And what you can hopefully see, it's a bit difficult on this screen, is that in the pre-stage you see very little difference between the diabetics and controls. Post, you don't see very much either, but that 3-hour time point later, we have this infiltration of immune cells. So, here, this is what happens when you steal pictures of the internet. It's been, there should be a picture of Frederick Vasily there. But I was going to say that this increase in its inflammatory tone, we'd already been suspecting for a long time. So when Frederick Vasily was in the lab in Stockholm with us, he did a study, but this is again now just in the resting state, where he looked at the phosphotranscriptome. So he could see that there was an increase in phosphostat-3 and phospho-react-2 in people with type 2 diabetes. And those of you who are not into JAK-STAT signaling, you just have to take my word for it. That's kind of an indication that there is some increased inflammatory tone in the basal state. It couldn't be seen in the mRNA, we couldn't see it there, but here we're looking at the phosphoprotein. So, and I guess that only showed you the men. So at the moment we're trying to study exactly the same thing in women, but we haven't quite got that far yet. But during the same time, another colleague in the lab, Lucille Dolay, was interested in using the adipose tissue. So she took the adipose tissue from exactly the same people. She studied both men and women. And so the interesting thing here was the adipose tissue, it's next to the exercising muscle, but it didn't do any work. So it's sort of an innocent bystander, but we wanted to see, was something happening there too? And to sort of cut a long story short, what we saw was a very, very similar difference again. So now I'm just showing you the pre versus post. So here's when in the adipose tissue, the pre versus post, when immediately no one got the bike, there's about 500, 600 genes that change in adipose tissue. But when you look at that 3-hour time point after they got the bike, here's the type 2 diabetes circle again, and it's much bigger than in the people with normal glucose tolerance. The adipose tissue is also responding very differently if you happen to have type 2 diabetes and get on the bike. So, now I go back to the muscle. There. Get this to work for me. Okay, it's just a reminder that's what it looked like in the muscle. So what Lucille did is she was thinking that perhaps what we should really look at is secretory factors and look at what's the tissue prostral. So here we have again the same data pretty much of the difference, but now she's only looking at the genes that encode for a secreted protein. So again, this is all kind of bioinformatics. She's taken all the genes and kind of looked at them and see what's the signature, and do any of these genes in the adipose tissue look like they would be secreted? And the little pizza pies up here shows that it's about the same in a normal glucose tolerant type 2 diabetics. So here is not, that doesn't explain the big difference here. But she was interested in seeing what's adipose tissue releasing in response to exercise. And there are a number of proteins that have been reported to do that. So we saw sort of the usual suspects, which is always nice. It means that you see what other people see. But what Lucille also found was a number of genes that hadn't really been studied that much before. And in particular, I want to tell you a little bit about this one, which is called oncostatin M. And as you can see, it goes up very, very markedly in the adipose tissue at 30-minute time points. And it comes back down again pretty much. So oncostatin M was the thing that we really were interested in studying. And why did we just pick oncostatin M? It wasn't completely random. What we already knew was that if you treat obese mice with oncostatin M, you actually have improvements in adipose tissue inflammation and insulin resistance and a lot of good things. Basically, that had been shown by other people. However, there's a bit of a difference in mice. The oncostatin M is increased specifically in skeletal muscle fibers. Now, because we have the data from the muscle, we knew that in the humans, oncostatin M does not increase in muscle from humans, but it does seem to increase in adipose tissue from humans. And indeed, we then went on to say, okay, the important thing is, so far we'd only looked at the mRNA in an adipose tissue. So what really happens, of course, if we then look to see the business end, we went to the blood samples to see is there really an increase in the circulation. And indeed there was. And interestingly, the increase in the circulation is higher in the normal glucose tolerant people than in type 2 diabetics. And we also saw that there was a sex difference in the response here. So what could it be doing? Well, of course, it's quite difficult to do the mechanistic studies in humans, but we tried to kind of figure out if we took cultured adipose tissue in vitro and we exposed them to a number of different substances, but one of them was oncostatin M, and we could see that we had a quite dramatic increase in phosphorylation of ERK. Now, phosphorylation of ERK can do a lot of things. One of the things it can do is increase lipolysis. And in an experiment I'm not showing you is that this actually does increase lipolysis. Oncostatin M increases lipolysis in cultured human adipose cells. In vivo, the only thing we can do is really go back to the tissue and see, okay, if people exercise, does ERK phosphorylation increase in adipose tissue? And indeed it does. But this, of course, is a little bit hand-wavy. I'm just saying it's correlated, but at least it goes in the right direction. We then went to try and think a bit more carefully. Like I said, we knew that oncostatin M doesn't come from muscle in humans. The other thing that we could see when we now went to the online databases to download all the different kinds of single-cell sequences is that oncostatin M is primarily expressed in immune cells. It is not expressed in adipose tissue. So again, we think it's the immune cells that might be coming in to do this. However, the receptors are specifically enriched in the adipocyte fractions. So what we think is happening, and also then we went to see, is this really true? So I'm showing you here some RNA scope
Well, basically, again, in adipose tissue slices, we specifically tried to go in with RNA to see whether we have CD68 as markers of immune cells, and see whether it actually co-localizes with the oncostatin M. And we think that monocytes and macrophages are the likely source of oncostatin M in human adipose tissue. And here's just to prove to you that in the muscle, we really see no oncostatin M at all being expressed. And the receptor goes up a little bit. So, let's skip that one. So what we think is happening is that when you run, you have a signal from your muscle that somehow recruits immune cells, not only to the muscle itself, but also into the adipose tissue, and that this is different in people with type 2 diabetes, and that alters metabolism. So how can you test this? Well, you can't test it in a human, so we tried to do sort of the next best thing. So we took some cultured immune cells, and we treated them with things that we thought would mimic exercise, like clenbuterol for scoring. And what we can see then is that when we do this to the immune cells in the test tube, we can have an increase in oncostatin M. The other thing that we did is we took cultured human muscle cells and we contracted them in vitro, so kind of see whether they produced something. And so we took this media from the contraction, and we contracted them with electricity, and we condensed them, we did a peak of the oncostatin M. So we think that there is the possibility of crosstalk. So what we think is happening then is that the exercise induces inflammation in adipose tissue and muscle, and somehow speaks to this factor, increases something in the immune cells, and the immune cells increase oncostatin M, and the oncostatin M change the adipose tissue metabolism. So that's our attempt at looking at all of the crosstalk. Now, before you completely fall asleep, I'll tell you a little bit about exercise-induced metabolomics. And so metabolomics was measured, we measured it both in the plasma, but I'll tell you here what's happening inside the muscle. So now we're back in muscle, in the muscle tissue. And what we then basically looked at untargeted metabolomics, so all the metabolites that change. And there's lots of them. So I'll just walk you through to kind of try and see some patterns. So here it's the female normal glucose tolerant in response to an exercise bout. Especially what you see is branched chain amino acids, you see lysophospholipids and sphingomyelin. So now we're in normal physiology. These are normal glucose tolerant people, and they are women. And that's what happens when you cycle on a bike. If you're a man, it's pretty much the same. Doesn't seem to be a big sex difference. Again, the branched chain amino acids are the biggest thing that happens, and you have a few changes in fatty acids that you didn't really see in the women. Now, type 2 diabetic women. It's a little bit different. We do see the branched chain amino acids. That seems to be like the number one thing that happens to metabolomics when you exercise. But we also see some changes in fatty acids in long chain PUFAs and sphingomyelins. So that's in type 2 diabetes. And in the men with type 2 diabetes, branched chain amino acids again, but now you have changes in lysophospholipid, diacylglycerol, and then these long chain fatty acids. So I'll just try and summarize it a little bit. So what happens all the time is branched chain amino acids. I didn't show you the TCA cycle, but most acyl conjugates, these things seem to change in muscle when you exercise. Disease-related difference are diacylglycerol and a few glycolysis-related metabolites. And sex-related differences are the ones that are more related to lipids, like long-chain PUFAs, lysophospholipids, and sphingomyelins. But I want to show you this look at it as well. So now here's one of my colleagues plotted this, sort of the change as a factor of time, because we have the before getting on the bike, getting off the bike, and afterwards. And so you see the amino acids have this kind of peak. It's just grouped on a number of different things. So here again, it's the women who have like a peak in branched chain amino acids, and then it kind of stabilizes. Let's add the men. The normal glucose tolerant men are very similar to women. You see this change in energy, sensing, metabolites, but there's a difference in lipids between men and women. Now we have the type 2 diabetics. So again, the women, we still have this amino acid change, but we have a much more sluggish response on the lipids. And now we have the type 2 diabetic men. And again, we see that the responses are sort of similar, but they are delayed. And we're still sort of trying to put our heads around this and get this all together. But I think, if you remember, the glucose challenge that I showed you in the beginning is often delayed in a person with type 2 diabetes. And what we seem to see is that it's not just glucose. The whole metabolism seems to be more sluggish. And now we're not going in with a glucose challenge, we're going in with an exercise challenge. And for some reason, the whole response just seems to be different. So now I want to kind of highlight a few things that, for those of you who kind of born with me here now. So this is an ongoing project that we're working on at the moment. We sort of call it multiple omics in metabolic disease, and it's just one single exercise bout. And we've just now started analysing things like DNA methylation. Now, the beauty of the study isn't so much that it's complicated. Of course, we've done every single omics we could think of, which is clearly expensive, but really the importance of the study comes from a very well phenotyped intervention. And this sort of intervention needs to be done not just in one population, it needs to be done in several populations. And perhaps if one can identify what are the key things we need to look at, then you don't have to do all the omics, but you could do the smart intervention in a different population to say, well, is this true? This conclusion that we want to say, which happens to be true for men and women who are about 57 years old and live in Stockholm, how does that actually pertain to the rest of the world? So, now for those of you who were just wondering what I was talking about, the take-home message is that an exercise bout in a person with type 2 diabetes results in a very different response than exactly the same bout in a person with normal glucose tolerance. So that's the one thing you need to write down. And the other thing is that many of these things are not different in the basal state. We know, like I said, we know for 40 years that muscle responds differently to insulin. But when you compare muscle biopsy from people with type 2 diabetes or normal glucose tolerance in the basal state, when they're just resting or fasting, there are very few differences that you could see, especially in things like the transcriptomic, which is what we've been studying for a long time. But you stress the system and you can unmask the differences. So, looking ahead, so now we come back to, is physiology important? So what I've tried to walk through is that we can now study on a global omic level things like DNA, we can study DNA methylation, histone methylation, with of course RNA or mRNA transcriptomics, which is something that 20 years ago was very, very expensive. Now this is pretty much routine. It's quite inexpensive to study this. Same with DNA methylation, that is quite inexpensive. However, studying protein and protein modifications is very expensive and very complicated. You need high sophisticated machinery, same thing with metabolomics. And then in the end, you need to have clever bioinformatics. But, so this is where I'd like to kind of come in. Even with the most sophisticated and most fantastic proteomic studies or protein modifications, or the most sophisticated single-cell RNA sequencing, in the end you end up with a list of correlations. And that list of correlations doesn't mean anything until you've translated it into physiology. Because otherwise it's just correlation. So the translation to physiology will require physiologists. And while I think that AI might help us with the bioinformatics, it will still only give us a more clever list. And if we cannot, as physiologists, test the hypothesis, then it will be meaningless. The other thing is, of course, we need to look at the different tissues. And finally, we like to think about this thing of precision medicine. So precision medicine is clearly something that is important, because we know that it's not one size fits all for a pill. We know it's not one size fits all even for exercise. I alluded to that at the start. And we know that for some people, maybe it's a cycle bout that's really efficacious, whereas for other people, it might be that strength training is more efficacious. But we cannot do precision medicine if we don't have multiple different populations. And at the moment, we are, as a sort of, collective, most of the data is from Northern European men. We're not even studying women well enough, and I think Professor Raveley highlighted that too. But we are only studying one small subgroup. And so I think it's really important to see, is an exercise bout in a man in Stockholm the same as an exercise bout in a man in Japan or an exercise bout in a man in Nigeria? That we don't even know yet. And so what I'd like to say at the moment, the low-hanging fruit isn't to do all this exactly the same. The low-hanging fruit is to figure out what is the key question, what is the key readout that you have found? And can you replicate that, or is that different? The other thing that, of course, needs to be done, and one thing that we've been studying is that an exercise bout done in the morning for somebody with type 2 diabetes is less efficacious at reducing blood glucose than one done in the afternoon. But that's only for one type of exercise. We use high-intensity interval training. Maybe if we had used another exercise intervention, we would have seen something different. And then the only readout there was glucose control over the next 48 hours. So that's not a particularly expensive measurement and can be done everywhere. The difficult thing is in careful physiological phenotyping, in the selecting of patient and the matching of patient, in making sure that the exercise intervention is exactly what you want them to do. And that, I think, is something that really can be done even on smaller budgets. It just takes very passionate physiologists to do it. So, even though I think looking ahead, we are moving towards larger omic analysis, but the large omic analysis doesn't have to be done all in one lab. It can be a collective, be it in rodents or in men. And to gain precision, for precision medicine, we need very much to increase the variation of the samples we study. We can't just be studying medical students in Boston. And the future will absolutely require collaboration, because not one person can do all this, not one lab can do all this. And again, I just want to thank all the lab that I share with Janine. And I was looking at the picture this morning, and we actually have people represented from every single continent, although at the moment the African subcontinent is represented by somebody from Egypt, so he looks a bit pale. But, so with that, asante sana. And promise will tell me when I should sit down.
There we go.
Yes.